Good morning, children. Yesterday we have learned about the autotrophic nutrition, heterotrophic nutrition, and in the heterotrophic nutrition there is types of parasitic nutrition and saprophytic nutrition and holozoic nutrition. And we learned about the process of photosynthesis. Photosynthesis is the process the green plants which prepare the food materials by using the inorganic compounds like carbon dioxide, water in the presence of light, but in the chlorophyll. So this is the equation 6CO2 12H2O in the presence of light and chlorophyll. They are forming C6H12O6, that is glucose, and 6 molecule of water and plus 6O2 is released into the atmosphere. So this is the glucose. So glucose is prepared by the process of photosynthesis and it is stored in the green leaves. So now we are going to test whether the leaves they contain starch or not. So starch is the another form of the glucose. Several glucose molecules they together form the starch. Now by doing the simple activity we are going to test the starch. So presence of starch in the leaves. So this is the arrangement of operators. For conducting or doing the experiment, each and every experiment, we have a aim. So what is our aim? Presence of starch in the leaf. To test the presence of starch in the leaves. What are the materials required? Test tube and beaker, boiling water, methylated spirit, leaves, and these are the leaves, thin leaves and asbestos gas, tripod stand and Bunsen burner. So these are the materials required. And before going to procedure, so we are testing the leaf. We are taking the leaf and we are boiling in the water and then we are boiling in the methylated spirit. So when we boil the leaf, what happens? Generally in these days, corona days, we are taking the bath with this neem leaves. When you boil the neem leaves, ultimately you observe that after some time, so the water gets green color. What happens means whatever the pigments are present in the leaf, they are mixed in the water. So they are dissolved in the water. So likely, and when you boil them, you observe that they lost their structure. So they become very delicate. So they are leaving, so that pigments are dissolved in the water. So we are using the methylated spirit in this to dissolve the chlorophyll pigments. Okay, chlorophyll pigments dissolve in the methylated spirit. We are taking the thin leaves and coming to our procedure. So we are taking a beaker which containing the boiling water. If you observe the operators, we are not directly eating the test tube. So we are not directly eating. Why? There is a reason is there. So boiling water. On the boiling water we are heating on the asbestos gas. And here we are taking the tripod stand. We are taking the tripod stand. On that we are placed asbestos gas. Above that we are placed the beaker which containing the boiling water. And we are taking a test tube. In the test tube we are taking the methylated spirit. And in that we are adding the leaves which are we are taking the thin leaves and that should be washed thoroughly and we have to clean it and dried with the help of tissue papers and then we place in the methylated spirit. So why we are not eating the directly? Because you know that the methylated, we are saying it is a spirit. It is volatile in nature. It may cause the burning. So to avoid that, we are using in the test tube. And if you observe the arrangement of the operators, so now it is tripods and asbestos gas heated and then slowly they transfer the heat to the beaker. So this boiling water, first boiling water started heating and then they eventually supply the heat. That heat helped to boil this methylated spirit and in that our leaf is going to be boiled. So while after some times we are doing after some time, it takes so much time because it has to first water should be heated, then after this that heat to be carried to this test tube, inside methylated spirit heated, and when it is heated, gradually our leaves turns into white color, pale color, colorless condition, and this solution turns into color, green color. So that indicates that our chlorophyll pigments which are present in the leaf, they are dissolved and they are mixed with the methylated spirit. So here what is the purpose of using methylated spirit? To dissolve the chlorophyll pigments. After some times we will take this leaf. So it is very careful that with the help of brush only we can take this leaf, because after heating, after boiling, it will become more delicate. So we will take that leaf into the petri dish and we will place it and we add the few drops of iodine. So by adding the iodine, this turns into pale color, white color turns into dark bluish black. Bluish black. So this is the, if starch is present, then only it turns into bluish black. Otherwise it won't respond. If you take the leaf of so many days, if you keeping the leaf in the dark room and if you collect that leaf and if you do the same experiment, you won't get the bluish black. If starch is present, then only it gives the bluish black. So by observing this color, we confirm that starch is present in the leaf. So where we are getting the starch? By the process of photosynthesis only. So in the photosynthesis, they are preparing the starch and the starch is present in the leaf. And what are the observations? So observations means when we add the iodine solution, so the leaf turns into bluish black. So inference are final, our result is, what is the result? So starch is present in the leaves. So that's why with the iodine test, the leaf turns into bluish black. Once again, if you remember the arrangement of operators, then it is very easy to write down the procedures. So just simply we say that arrange the apparatus as shown in the figure, and take a tripod stand, arrange the asbestos gas. After that, put a beaker which containing the boiling water, and in that place a test tube. In the test tube we are taking the methylated spirit. Gradually when we heat the when we supply the heat, gradually this water boils and that heat is eventually supplied to the test tube, and that helps to boil the methylated spirit and whatever the pigments are present in that, they become dissolved in the methylated spirit. They may ask questions anyway, so they may miss some points. And what is this? They may ask in 1 mark question. And why we are using the methylated spirit? Methylated spirit used to dissolve the chlorophyll. Okay. So this is the experiment or activity, presence of starch in the leaves. Okay, children. So in that experiment, that leaves presence of starch, we are observed how we are removing the chlorophyll pigment by using the methylated spirit. Some precautions we have to follow that. We should not eat directly methylated spirit because it is volatile in nature, and and it should be boiled in the only water bath only, not directly. We should not heat directly. And next topic is materials required for photosynthesis. Materials required for photosynthesis. What are the materials required for the photosynthesis? If you observe the equation, you see that you may simply say that carbon dioxide is necessary, water is necessary, all these things. But some scientists are there, so they try to work on it. And in them, one of the scientists is von Helmont. What is the name of that scientist? Von Helmont. So he thought that So plant mass is increased, plant body size is increased due to the uptake of water. So water is the main reason, water is the main reason to increase the plant body or plant mass. The size of the plant is increased due to the water. But at that time they don't know the process of photosynthesis. Actually, plant body mass is increased due to the photosynthesis. Photosynthesis, they prepare the food materials, those materials used for the releasing of energy, and they help to the growth of the plant. So this is the one element. He said that water is responsible for the plant growth. This is studied in the previous 6th class like that. And next, another scientist, uh, Priestley, he want to prove that the relation between the air and photosynthesis. Air and photosynthesis. What is the relation between the air and photosynthesis? The scientist's name is Joseph Priestley. Joseph Priestley. So he discovered oxygen gas. We know that who discovered the oxygen gas? Joseph Priestley. So he want to know that he did lots of experiments. In that one of the experiments he want to prove that relation between the air and photosynthesis. So to prove that, whatever the experiment he done by he used the bell jar. So he used the bell jar. So we say that the bell jar experiments are done by the Joseph Priestley to know the role of air in the process of photosynthesis. So air role. So simply he take the bell jar and simply he take the bell jar and put a burning candle. So put a burning candle. First he did that, he take a bell jar. So he lighting a lamp, lighting a candle, and on that he put the bell jar. So he observed that after some time, so what happens? This is turned off. So due to lack of oxygen, so whatever the carbon dioxide, whatever the oxygen is there, that is utilized by the burning candle, we know that, and it is releasing the carbon dioxide. So with that carbon dioxide, it won't light it. So it is turned off. And next, in this experiment only, he introduced a small mice living. Okay, small mice. So when he put this candle, at that time also, after some times it turned off, and when he put the mice, at that time also he observed that it is also dead. What is the reason for that? So due to lack of air, so due to lack of oxygen, it causes the saponification and it leads to the death of this mice. And so next he placed a small mint plant. He placed the small mint plant. So when he placed the small mint plant, so he observed that. Next what he placed? He placed the mint plant. So this all the setup is there in the outside the sunlight. So when he placed the sunlight, this is the glass containing bell jar. So ultimately it takes the plant, of course it performs the photosynthesis. It takes the sunlight and everything, it performs the photosynthesis, and byproduct of the photosynthesis is oxygen gas. It releases the oxygen. Oxygen is, this mint plant is releasing the oxygen, and one side, another side, the burning candle and the mice, due to respiration, it is releasing the carbon dioxide, and burning candle, it is releasing the carbon dioxide. Whatever the carbon dioxide is released, that is absorbed by the mint plant. All these things are not known by the Priestley. At that time, he does not know that. So, mint plant really absorb the oxygen, absorb the carbon dioxide, and this releases the carbon dioxide, and this mint plant releases the oxygen. They don't know all these things, but by doing this simply experiment, he want to concluded that some harmful gases which are released by the burning candle and mice, they somehow damage the air. That causes the first time before placing the mint plant. So some gases are released in the air, they damage the atmosphere of the bell jar, that causes the death of this mice and candle, turn off the candle, exhausted. And next when he placed the mint plant, he concluded that so this mint plant, the harmful gases which are released by the burning candle and mice, they are restored in the plant. So plants restore the air what the breathing animals and burning candles remove. We know that that gas is carbon dioxide that is stored by the plants, and for the process of photosynthesis they are releasing the oxygen that is utilized. So this is the experiment did by Joseph Priestley. This is the one of the milestone for the process of to know the more about the photosynthesis process. So once again, and Joseph Priestley, he discovered the oxygen. He did not name the oxygen. Then Lavoisier, the scientist Lavoisier, he coined the word oxygen. Oxygen name is given by the Lavoisier. Lavoisier scientist coined the name of oxygen. So bell jar experiment is done by Priestley. So with this experiment he want to prove that some harmful gases which are released in the bell jar, that is burning candle and mice released, those are restored in the plants. He also confirmed that gases exchange was going on. Gas exchange was going on and plants were giving out the gas that supports the burning candle and essential for the survival of animals. So whatever the gas is required for burning candle and mice, that gas is released by the plant. We know that in the plants there are minute pores are present in the plant's leaves. They are known as stomata. Stomata helps for the gases exchange. And not only stomata, on the stem they have the lenticels. Lenticels also used for the gases exchange. Okay children? So materials required for the photosynthesis in this topic. So some scientist who Van Helmont, he thought that the plant body mass is increased due to the water taking and role of air in the process of photosynthesis. Further, Joseph Priestley did experiment with by using the bell jar, bell jar experiment, and finally he concluded that some harmful gases, gases which are released by the burning candle and mice, they are restored in the plant. Simply we can say that oxygen is released and they releasing the carbon dioxide during the process of respiration, that is restored in the plants. Okay children, thank you.